Hi, Monica. How are you? Um, the microphone, so you can unmute yourself, is on the bottom right hand. There's a little um, microphone button. So, yeah. Can you hear me?
Yes.
Okay, great. Can you say something again to test if the sound is good?
Hello. Is it
Uh,
audible?
it's a little bit quiet, but it could be me. So, are you close to your microphone?
Yes, I am.
Okay. Okay, good. Maybe it's just my reception. Oh, we'll open the room in like around 4 minutes, 4 to 5 minutes. So,
Yeah.
and then I'll introduce you. And yeah. So let's wait for people to arrive, and then we'll
Okay.
start.
Yeah.
Thank you.
Yeah.
Hello, everyone. Thank you so much for coming, everyone in the audience. We'll start in a few minutes. So stay tuned. Thank you. Hi, Serena. How are you doing?
Hello. Hi, Monica.
Hi.
Really looking forward to this topic today. Sounds so fascinating.
Yeah, me too. Oh, it's really great work, Monica. Hi, Gilbert. How are you doing? Meet Monica.
Hello, everybody. Hi, Monica.
Hello.
Hey, everyone.
Okay, it's 12 p.m. EST. We can slowly start. I'll introduce you, and then I guess people will continue coming in. But yeah, I think we can start. So, welcome everyone to the Science Society. Thank you for coming. We have another day and another really interesting and very impactful work that will be presented by our guest speaker. And let me introduce you to her. Monica Priyadarshini. She will be introducing, talking about her work that was published in Nature Methods, and she is a biological and environmental scientist and and in the engineering division at King Abdullah University of Science and Technology in Tuwal Kingdom of Saudi Arabia, and she did she's in the she's there doing her research in transgenerational gene silencing and epigenetics, and this work was very recently published. And as I said, it's a very impactful and very important publication. And I'll let Monica tell you all about it first, and then I'll continue why I think it's so important. And so, yeah, thank you so much, Monica, for being here and for taking the time to share your work with us. And yeah, the stage is yours.
Thanks, Katerina, and I'm thankful for making me a part of this science society. I didn't know about it before, but I think it's a great platform to share research and give a general overview of the work that we are all doing. And I'll just briefly introduce myself, even though you already did. My name is Monica, and I come from India. Uh, and I hope everyone can hear me. If you cannot, just let me know. Unmute your mic and let me know, because I feel like sometimes it's showing that the connection is unstable. So at any point, just let me know. Anyway, so I come from India. I shortly was in Germany for a short project where I got interested in epigenetics at Max Planck Institute for Biophysical Chemistry after my master's. And then I took up a PhD position at King Abdullah University for Science and Technology in Saudi Arabia, um, following one of the professors who joined here actually. So I didn't know about this place before, but turns out that I came here to do some really interesting research, and I'm currently just in the process of moving on to my next career step. And currently still here as I mean, I've graduated from my PhD and just waiting for my visa process to be done. So anyway, today I'm actually going to talk about my work, which was one of my main projects during my PhD. And so this is my paper, first author paper from the work that I did. And just to give a brief overview about my research, so there's a model organism that I work with, and it's called C. elegans. It's a round worm, and it's one of the most famous model systems that people have used, other than fruit flies and mice and rats. It's one of the more fundamental model organisms in the sense that there are a lot of biological pathways which are uh, conserved from humans and mammals to worms, and that's why it makes it very interesting for us to study a more simple organism, especially developmental pathways in a simple organism, and then be able to kind of make a correlation with higher organisms. And there are a lot of interesting science that at this stage that we can make use of these simpler organisms to study, which we cannot do in higher and more complex organisms. For example, transgenerational inheritance, or for that matter, any inheritance studies. If you had to do a generational time point in rats or mice, it would not be possible by a single PhD student. Would be a very long PhD project. But for C. elegans, this worm is very interesting because it's a hermaphrodite, so all its progeny is clonal, so to speak. And also, its life cycle is 3 days. So every 3 days, you have a new generation of worms that you can study. So this was one of the main advantage of doing this epigenetic transgenerational inheritance study in worms. And then this whole field of transgenerational inheritance was something that was discovered in C. elegans. So apart from winning, 2 or 3 Nobel Prize awards by this worm, it has many interesting features which now people are trying to study in even higher organisms. So what's transgenerational inheritance? I would like the audience to even understand what that would mean is when, let's say, there's an environmental trigger of some sort, and the parent is exposed to some kind of environmental cue, and it has some response to it. But this response can get inherited not just in one generation, which is their progeny, but even in their grandchildren, and then so on and so on, who were not all these progenies in the later generations that were not exposed to the initial stimuli. This is what we call transgenerational inheritance. And this was specifically first observed in worms when through a method called RNA interference, which was one of the Nobel Prize winning work done in C. elegans and by 2 famous scientists, Professor Andrew Fire and Professor Mello. So what happens in worms is they have a very strong, let's say, defense mechanism which is based on small RNAs. And these small RNAs can recognize any invading, so to speak, complementary RNAs, and then once they degrade it, they make a memory of these invasive RNAs, and if they encounter it, then they will always have a population of these small RNAs which gets carried on to the next generation, and they can go on silencing it. When RNAi happened, then people started to understand what this whole small RNA dependent phenomena is, and that it can go from one generation to the next to the next. So then one of the main questions that I asked during my work is, well, there are more than one kind of small RNAs in worms, and one of the more important ones that we were interested in are called piRNAs, or Piwi-associated small RNAs. And why was this important? Because these small RNAs are more enriched in the germ cells of the worms. So that means that if you have any thing happening in the germline, that it would, these piRNAs are known to protect the germ cells against some detrimental effect of repeat-rich elements like transposons, retrotransposons that can affect the fitness of the next generation. So they do have a protective defense mechanism. But there are several other functions of piRNAs in worms that we are not aware of. And we wanted to explore more what these piRNAs could do, and essentially use it to make a tool that can be used to manipulate things only in the germline. So we wanted, and another thing that I would like to highlight is through RNAi in worms, there's only a handful of few endogenous targets that people have shown can be silenced. And if you silence, for example, a gene which is required for embryo fertility in C. elegans, if you target this gene, you can silence them for a couple of generations, but that's the only known endogenous gene which can be silenced, and that silencing is inherited. So another basic question that we had is this phenomenon specific to a few handful genes, or is it a general phenomenon? Or or what is it? Are there other endogenous targets that we can silence and then see inheritance? Because it would be a much better platform to study transgenerational inheritance if we can look at more interesting genes that can be silenced, but also that give a robust phenotype upon silencing, and that phenotype can be, used for quantifying uh, the silencing process. And you can imagine if you are silencing any genes in the germline, that the silencing can cause very severe or lethal effects because it's in the germline. So there are very few genes that you could actually target and see a real phenotype instead of, something like death or embryonic arrest or things or sterility. Um, so there are two of many genes that we targeted, and these genes fall under a category called HIMS, or high incidence of male. And C. elegans, like I said, is generally a hermaphrodite, but you can have, in rare circumstances, males. So there are two genes, HIM5 and HIM8, that we chose, and both these genes are involved in the chromosome segregation pathway. And if you knock out these genes, then you start seeing an abundance of males on the plate along with hermaphrodites. So this is one of those phenotypes that you can actually are healthy, and it is something that you can quantify. And so we thought that these would be great targets. And even though RNAi is a very well-established method of silencing genes in the field, um, these two genes are not easily silenced by RNAi. And we don't fully know why. It's possible that RNAi doesn't work very well in the sperm, but might be more good in, the female germline. So we thought that piRNAi could be a better way of silencing genes, especially because it's enriched both in the male and the female germline. And so first we targeted one of the genes, HIM5, using short piRNA sequences. And to tell you a little bit about piRNAs, in C. elegans, the piRNAs are 21 nucleotide sequences, and they have a bias for a 5' uracil. So on the 5' they have a U, and then you have 20 base pairs of um, of uh, any other sequence that it's targeting. So what we thought as a tool development, what would be interesting, and what we thought could be similar to CRISPR, how you have today we have CRISPR where you have these sgRNAs that target Cas9 to a particular loci in the genome to do gene editing. We thought, oh, can we have a similar system in worms where we use it to manipulate its endogenous pathway to go and silence genes. So the piRNA sequences, like I said, are 20 base pairs in size, and what we did was we selected like a cluster, a 1.5 kilobases large endogenous clusters, which have at least 6 different endogenous piRNAs, and without manipulating the 5' U, the next 20 base pairs is what we replaced with exonic sequence of the gene that we wanted to target. And with that, we created like a synthetic piRNA cluster, and then we simply injected these clusters into the gonad of the worm, and with a few other co-injection markers to know if you get transgenic animals. And then we looked at whether we observe any phenotypes or not. And so what we did see that for the gene HIM5, which we tested by doing RNAi or by doing piRNA interference. This is what we called it actually. Since we are using piRNA to target genes, we call it piRNAi. And we compared the male frequency of males in the population from both these methods, and we saw that we got at least 40% males in by using piRNAi mediated knockdown versus RNAi, which we found was something really, really cool. And we're like, oh wow, this is great. Uh, but but can this be inherited? And one more reason for doing this was also that in our field, people have also depended a lot on transgenes. So one of the things on which gene silencing would be, gene silencing studies have been done a lot in the germline is just by silencing a GFP, a germline GFP. And that uh, is, as you can imagine, a GFP is an is a transgene. It's a gene which is not in it's not an endogenous gene for C. elegans. It's something that we have uh, improvised on to express in a different organism. So I think small RNA-mediated defense mechanisms are they very easily recognize transgenes, and so when they silence a transgene, it can be silenced forever, right? Because they look at it as a foreign DNA, as a foreign element. And so we think if we want to really study endogenous uh, pathways, and if we want to understand epigenetic inheritance in true sense, we have to target something which is endogenous and not something that doesn't belong to the organism. And that was one other motive to develop a tool that can robustly silence germline genes and then study whether these silence genes can undergo inheritance. So the next step obviously was to test whether piRNA-mediated silencing of HIM5 shows inherited silencing. And so we tested this by picking, in every generation, we would pick animals at an early stage, which cannot mate with males. So one of the things that can happen if you have males and hermaphrodites on the plate, then if they are at a mating stage of their life cycle, they would start mating, and that could induce uh, a wrong count of males that you would see on the plates. So to avoid any of these problems, we would pick animals at an early stage, life stage, where it was impossible for it to mate with other animals, and we would pick them and then look at the progeny of those animals and see if we have any male population. And what we observed is for the gene HIM5, the inheritance lasted for 3 generations after the initial trigger was removed. We would completely remove the initial trigger, and the way it can be done is what we inject is a DNA, and it makes a high copy number array, which we call extra chromosomal array. So if you have a selection for the array, you can maintain the array, and if you remove the selection, you can get rid of the array. And so we would select non-array animals and propagate them for multiple generations and see if the phenotype that we are looking for is still there. And I would keep doing the inheritance assay until I no longer see the phenotype. So it was a very interesting approach and also a very simple approach for for studying inheritance pathways. But then we started thinking of answering more fundamental questions about the piRNA pathway itself. We can use this tool not only to study germline pathways, but also to understand in depth how the piRNA pathway works. So some of the things that we tested is looking at other genetic requirements for piRNA pathway to function. And so one of the things which are important for piRNA pathways is these small RNA pathways depend on a particular group of proteins that are called argonaute proteins, and their main function is to bind a particular group of RNAs and then go and bind the target uh, mRNA sequence and then cut it. So this is how they work. And all argonauts uh, generally have this function, and this is conserved from worms to flies to mammals. Um, so what we wanted to look at is now we've developed a tool called piRNAi where we are reprogramming and designing synthetic piRNAs and injecting into the animals. Are they also working via the endogenous piRNA pathway? So when we tested that, in worms we have a lot of mutants where different genes that are responsible for a particular small RNA pathway have been knocked out. So we can simply test piRNAi in those particular mutant backgrounds, and so we did that. And for the piRNA pathway, one of the most important argonaute proteins is called PRG1. That is its fundamental binding protein. So we observed that in the PRG1 mutant, if you inject piRNAs against the gene HIM5, you do not observe any males. So this was also a sanity check for us that, okay, whatever we have designed works via the piRNA pathway. And then we had also another bunch of genes that we then tested, mutants that we tested that could also be involved in initiating piRNA-mediated silencing of endogenous genes. Um, and then we found that mostly all the mutants could be involved in later stages of silencing where the silencing has been established, but they were not really essential for initiating piRNA-mediated silencing. So this tool also helped us to discriminate between two phases of silencing. One is initiation of piRNA silencing, and the other is maintenance of piRNA-mediated silencing. And then, there are a lot of other results which are interesting, but would broadly be something that the field would be interested. But the two main things that I would like to highlight from this study, which I found was quite interesting, is so I talked about it being epigenetic inheritance. So what I mean by that, epigenetic, the word in itself means in something other than DNA. So we know that most of the genetic inheritance means that it's DNA-based inheritance, but epigenetic means inheritance that require uh, non-DNA material. So that could be proteins, RNAs, and histone modifications, um, which are the nucleosome forming units around which the DNA modifies. So basically, without making any changes to DNA itself, there are other factors that can be required for inheritance. That's what we call epigenetic inheritance. So some of the markers for epigenetic inheritance that has become widely popular are histone modifications, DNA modifications, and also small RNAs and long non-coding RNAs. And so one of the things that we usually look at, which is a marker for gene silencing, is a very prominent histone modification, which is on histone H3 lysine residue 9. It's called H3K9 trimethylation mark. So if any location is silenced, then there is a method called chromatin immunoprecipitation, where you look at the DNA in DNA in its chromatin form, where it is bound with histones, and you look at the modification on these histones, and then identify whether that region of the gene is active or silenced. So we looked at what we call a CHIP signal, which is chromatin immunoprecipitation signal, and you can use specific antibodies for specific histone modifications. So in our case, we looked at H3K9 trimethylation modifications, and we found that if you the we found that the uh, you get very specific enrichment of H3K9 trimethylation at the target of your interest when you silence it using piRNAi. And this was also another evidence for us to show that the the silencing is epigenetic, and so probably the inheritance that follows is epigenetic as well. Um, but one more thing that we observed is that the silencing of this gene can actually spread into neighboring genes. This is something that we probably cannot control because we may have uh, we may have a target uh, for a particular gene, but sometimes in C. elegans there are some genes that are clustered. They are within something which is called an operon. So there are multiple genes within a very big sequence, and so it is possible that when you're targeting a single gene, you're basically targeting something adjacent to it also, and it spreads. But we didn't see any phenotype coming from the adjacent genes, but we did see reduction in the transcript levels of the genes in the operon. Um, and that's also kind of an interesting thing, but it's not an unusual phenomenon with silencing. Then one last thing that I would really want to highlight about my work, which was one of the most interesting things for me, um, and as a concept, we thought of things being as transgenerationally silenced, but what we also wanted to understand is what are the requirements for transgenerational inheritance of silencing, and can the transgenerational inheritance be shortened or reversed? Can the transgenerational inheritance become permanent? What under what conditions can we achieve these kind of um, uh, phenomenon? So what we wanted to do is look at certain genes that are responsible for transgenerational inheritance, and one of them is called HERD1. Um, and HERD1 was basically identified in a screen for studying transgenerational inheritance through RNA interference. And what we did was similar to when we were doing the genetic mutants. We injected piRNAs against the gene HIM5 and in the HERD1 mutant, and then we looked at the inheritance pattern, and we see that the inheritance doesn't really last beyond 1 or 2 generations, which basically told us that this gene is required for inheritance. In the absence of this gene, piRNA-mediated inheritance cannot happen. But what was also interesting is we wanted to know, okay, when can we make, can the inheritance become permanent? We know that we can prevent inheritance in certain cases, but can we elongate the inheritance? Can it go on for multiple generations? So for that, we kind of made use of another inducible mutant of PRG1. PRG1, as I said, is responsible for piRNA pathway. But what happens in the absence of PRG1, the piRNA pathway doesn't function. But we wanted to have a mutant where we can induce degradation of PRG1 and then study the effect. There have been previous studies which link that PRG1 can cause permanent silencing. There is previous literature on that, but it's unclear whether it's PRG1 or the maintenance of silencing which actually leads to permanent silencing. And it has been shown for transgenes such as GFP, so we were interested to know whether endogenous genes can actually undergo permanent silencing, because it would be counterintuitive for organisms to silence their own genes permanently. So is that even possible? So what we did was, there was a strain which we received from a lab of Andy Fire. It's a very interesting strain where the PRG1 protein gene has been tagged with a degron. A degron is another protein which basically, if it is bound to a protein of interest, it degrades that protein in the presence of a chemical or a drug, which is called auxin. So if you add auxin to the nutrient media, then it will be a signal for the worms to make PRG1 with degron, and the degron will actually degrade PRG1. So to do this experiment, we initially injected the piRNA, synthetic piRNAs, into this PRG1 degron strain without adding any drug, so that we start seeing the phenotype for HIM5, which was males. After that, we degraded PRG1 by putting auxin into the nutrient media, because then it would start degrading the PRG1 in the worms. And then we looked at the inheritance patterns. And what we observed was very, very interesting, that in the absence, once you established piRNA-mediated silencing and you got rid of the piRNA stimulus, and then you also degraded PRG1 and piRNA pathway, we observe permanent silencing of HIM5. And as soon as we stopped propagating the animals on the drug auxin, this whole permanent silencing phenomena got reversed. And then the inheritance became shorter, and it came back, it became non-permanent, so to speak. So the more interesting thing that came out of this last study was that the protein itself, PRG1, which is responsible for the piRNA pathway, is required to establish or initiate the piRNA-mediated silencing, but is also required to prevent permanent silencing. And it's very interesting that the same protein is required for both these processes, instead of just being responsible for one. And so this has opened a whole new, plethora of questions that we would next want to answer in the field, especially in terms of understanding fundamentally how the inheritance works. So this is in a nutshell of what this paper is about and some of its key results. I didn't go through all of them, but I only explained the ones that I thought were the most important. And I am happy to take any questions now.
Thank you so much, Monica, for your really great explanation. I'm sorry, my family is home, so there's some background noise. So, yeah, I think your work is so important, and I wanted to let everyone know why. Like, I worked in your memory for a while during my PhD and after, and I always was so interested on inheritance of epigenetic mechanisms and so on. Um, in the interest of learning how long trauma can persist or how it gets inherited. So I think your work will be so important to tackle those questions, and also editing in the germline, the way you do it, I think will open up a lot of stuff what I'm currently working on generating out of stem cells allogeneic transplants that can be you that won't have any rejection in other humans. I can't talk much about that because that's for company wide. I think it's important, but because it would just tell people what I'm doing. But yeah, so thank you so much. So I just wanted for the audience to give, the reason why I think this is really important work, and I was waiting so long for somebody to publish this basically. So thank you, Monica. And yeah, please, if you have any questions in the audience, raise your hands and flash your mics up here on the stage. And yeah, go ahead and ask your questions or comments.
Yes, Monica, fascinating, fascinating stuff. I'm a little unclear. I'm trying to piece together the details mechanistically. Could you summarize again what the mechanism of what you know of the piRNA suppression is and how it gets transferred? And I'm also curious in terms of the inheritance with multiple generations. You touched on it in some ways, but how in some cases it fades after a few generations, but in what's actually happening there? Is it
Yeah.
yeah?
So yeah, I didn't go into the detail of the piRNA pathway, but I can just explain a little bit. So in C. elegans, like I said, it has many different small RNA pathways, and they are really experts at recognizing foreign DNA or foreign material. So for example, in the case of RNAi, if you introduce double-stranded RNA complementary to the gene of your interest, then the RNAi pathway gets activated, and there are a number of organoid proteins which are going to form a complex to then degrade not only the double-stranded RNA, but it makes more of those double-stranded RNAs. It amplifies that signal, and those double-stranded RNAs become single-stranded. They find its complementary sequence in the genome, and then make more and more of the complementary short RNAs, and these RNAs will then recognize the transcripts. So every time the gene gets transcribed, you have the mRNA sequence. The complementary short interfering RNAs will basically bind to the mRNA of interest and cut it, and it will keep cutting it, and this is basically called post-transcriptional degradation or silencing. And what happens, and then there's what we call as the transcriptional silencing, is that this entire small RNA machinery with some other nuclear organoids that can go from the cytoplasm into the nucleus, they go and find the genomic location of your gene of interest and bring the silencing machinery over there, and then pause the RNA polymerase from making more transcripts. So it's more like a negative feedback. And you have other silencing modifications that are brought in, like the histone trimethylation marks, which are put on that gene of interest, and basically that locus becomes silenced. So
So,
this
so
is
like the RNA in action, it's actually replicating and acting on the transcripts of the complementary
yes,
sequence.
yes.
Oh my God!
Basically. And so the piRNAs are no different. They are very similar, instead that they don't require double-stranded RNAs as a signal. Inherently, in C. elegans, for example, there are more than 16,000 different piRNAs, but they rarely map to anything within the genome. So we know a little bit to some of the transposons, which are there in C. elegans, and they're not very much. But there are other sequences also, which probably were like over evolutionary time for other transposons and other things that it was targeting. So piRNAs are transcribed as single short 21 base pair RNAs, which find its complement in the genome. And then the entire machinery is somewhat the same in identifying the mRNA and cutting it, and then putting the silencing marks at the genomic level. So yes, there is
So
an
so over the
amplification.
generation, oh, I'm sorry. So over the
Yeah, go ahead.
generations, what actually happens for it to fade away? Is it its own replication cycle gets silenced, or?
Yes, so this is one of the hypotheses. We still don't know. We don't fully understand the molecular process of inheritance. And one of the theories that we have is these small RNA populations are basically moving from in. They are in the germ cells of uh, and they're passed on to the next generation. And over time, this acts as a, like a memory from the previous generation that, hey, you need to silence this particular gene because the complementary RNAs are present. But over time, I think that the organism also understands or remembers that this is my own gene. So there could be a mechanism to prevent the silencing process also. So over time, the small complementary RNA signal dilutes, and basically it fades out. And this is what I would think is the case for endogenous genes. But if you had something which is not belonging to the worm's genome, then that process might be different, I would think.
Fascinating. Um, do we want to ask other questions from the audience, or take questions?
Go ahead.
I was, it's very curious to me how the the alterations work themselves out as the generations go further. That was just really interesting to me. I didn't necessarily expect that to happen.
Yeah, and like I said, like epigenetics is like a combination of small RNAs, histone modifications, and these are things that is currently very hot in the field. Especially one of the things I spoke about was histone modifications, and one of the famous modification for gene silencing is H3K9 trimethylation mark, um, and how these non-DNA modifications actually dictate whether the gene is going to be expressed or silenced is very interesting. And not just that, these modifications then get passed on to the next generation uh, to to preserve the memory from the prior generation. One thing that I would like to add, which is not my work, but in the field, people are also looking at the role of piRNA pathways in inheritance of more of an environmental effect, like starvation, or uh, if the worm is invaded by a pathogen, how it remembers the memory and passes to the next generation also involves the piRNA pathway. And this is just to highlight that these small RNAs have multiple roles in defense, but defense mechanism, but also, you know, to to basically have a memory for the fitness of the organism itself.
It's curious how. Oh, go ahead,
No, no,
Katerina.
go ahead. That's fine.
It's so curious how a histone modification could be passed on. Is there some factor that is selectively um, recognizing a sequence and then modifying on the histone repeatedly, or is it being is that factor being transcribed, or I mean, what's going on there?
So one thing that we know for now, and it's a little bit different from higher organisms and mammals. In mammals, we rarely see transgenerational effects. It's now becoming more clear, but for example, one of the modifications is the DNA methylation. But what happens in mammals is usually when when the embryo, in the next generation, when meiosis happens and a new embryo forms, all the previous modifications are erased. It's like a clean slate for the new progeny, and then things are written back on it from the beginning. In worms, it's not the case, and we think that there, like I said, these small RNAs, if they get carried on from, let's say, the parent to the next generation of progeny, then those small RNAs would basically initiate the cascade of silencing, which would start by, let's say, the gene makes transcripts, forms mRNA. These small RNAs are going to go and target these mRNA transcripts. And then you will see degradation in the cytoplasm, but also then in the nucleus, the transcriptional silencing will initiate, and they'll put the silencing marks. So I think as long as you'll have these small RNAs, they are basically carrying the memory to initiate the silencing mechanism in every generation. This is one of the things I think is happening.
I see. So it's in essence, it's possessed, and that carries on in
Yeah,
the germline.
yeah. And
Fascinating.
I
And
mean,
go
yeah,
ahead.
and it has to be in the germline. Sorry. I was just emphasizing, like, if you have changes in the somatic cells, that's unlikely to be carried on in the next generation, right? Because it's just gonna stay
Mm-hmm.
in the cells that have already differentiated and are not going to be passed on. But if the changes happen in the germline, then it's likely that it's going to inherit in the next generation, because it's the germ cells from the parent that goes into the offspring.
Right.
So,
Yeah,
yeah.
and just to remind everyone, when pregnant woman, so the first epigenetic like big studies were in the Netherlands because there was this huge flood in the 50s, and the afterwards in the next generation they had a um, high presentation in the population of schizophrenia. So they looked at so pregnant women during that time that went through this immense stress and also scarcity because they were cut off of supplies for a long time. They saw like high increased rates in schizophrenia then. And just to explain, in a pregnant woman, relatively early on, there are three generations of germline. You have the mother, you have the child, of course, and then you have already the eggs built in the baby, basically, or in the embryo. So you have basically, if a big stress factor happens in a pregnant woman, you're affecting basically three generations, which is very like important to know. Combined with, just to give you like a real world impact, there came a study out last year that in the US it is 2 times, almost 3 times higher likely for a pregnant woman to experience trauma or even homicide than a woman that is not pregnant. Pregnant women gets beaten more often and killed more often, almost to a factor of 3 in the US. Like that was looked at the US, so this was very shocking data. But because of all of these just horrible facts that just exist in the world, it's really important to figure out those mechanisms, how trauma gets transmitted to the next generation, and then we will have at some point the tools maybe to silence them. I think we can save like the next generations of impact that trauma, for example, could have made. So but there's a lot of more fields where Monica's work will be very important. So again, just stating like how much real world impact Monica's work could
Yeah.
have in the future.
Actually, just to add on to what you said, Katerina, there was a TED talk a few years ago about behavioral studies and how that also affects the epigenetic environment. And I don't know if I would really encourage everyone to go and see, because that work was one of the few works that linked what we see behaviorally to what is actually happening in your DNA. And I thought that was interesting. I think it was studies in rats or mice, um, and what they show is the way the mom uh, rat treated its babies could be inherited in the next generation, and it was really environment dependent and not genetics, because they basically were able to discriminate that if the baby rats were given to a mother which was very caring versus a mother which was non-caring, it put different epigenetic marks in their DNA. And I thought that was a very interesting study, and also then it kind of shows the strong importance of epigenetic marks, that even if it's not, let's say, inherited, but from your environment, there are epigenetic changes that occur in your genome, and depending on what the signal may be, it may or may not get inherited. Nowadays, there are studies on obesity and metabolism, and there's definitely some things that people are looking at, parental obesity being inherited in the progeny. And we know that this is also one of the very important studies nowadays with nutrition and high fat diets. So people want to understand how much are the future generations succumbing to obesity if their parents were exposed to fat rich diet and things like that. And I feel that the worm is only scratching the surface. And the reason we can do these studies in these simpler organisms are basically because, one, these phenomena were identified here, but also, like I said, they have very short generation times. I can only imagine doing the study in 3 generations of humans would be a much longer time-taking process, and would probably not even be ethical at this point to do. But it's a way for us to find at least a scientific way of looking at transgenerational inheritance, and also I think piRNAi as a process, because I feel it's inducible. It's
Hello
not.
guys, hello everyone. Science is
Sorry, I think
I'm sorry, no, it was fine. It's, some
um
people just come to
uh,
disturb.
yeah, I
Sorry,
just
it's a public
it's
room,
fine.
you know, you never know.
I just want to touch upon the fact that piRNAi is also, let's say, inducible, and something that would be only persistent uh, as long as you have the array. You can have it, and then if you remove the piRNAi array, we may or may not see the effect of the silencing process. So anything which is inducible is in a way very useful, because you're not making any modifications to the gene itself, like you're not damaging the DNA, but you have an external way of controlling the gene expression, which I find is very, very useful and important to have. But again, I would also argue that this system is specific to C. elegans right now and may not work in higher organisms, but it does pave the path to find similar ways of manipulating genes in higher organisms.
Yes, I agree. I agree with you that ideally we have these, we use your type of method and not, changing really the code of DNA forever. So if it's something we can modulate ideally in real time, because changing, we never know, right, if it will lead maybe to cancer or to any other complication. I know that a guest speaker here that looked for safe harbor gene loci for modulating DNA, but it's still, I think, too high of a risk and also to get it approved. So ideally, we use your method in humans one day, and yeah, like we.
Yeah, I
That's why,
think
yeah.
yeah, I think recently RNAi was approved for drug discovery or something, and it's almost 20 years, uh, definitely more than 10 years since its discovery, and yeah, so it's going to be a while before these things would happen.
Yeah, I know. It's always in science like that, but maybe we don't live anymore to see it, but maybe we do. But
Yeah,
yeah, all we can do is try our best. Um,
yeah,
but yeah, I also used to work in studies where stress during development or by mom mice, what they can induce, or separation. We were kind of motivated on the current state here politically, that the illegal immigrant kids were separated from their parents. So we did the study of maternal separation, and
right.
yeah, epigenetic factors are quite important, and RNA level modulation. We saw a long-term impact on cognition, and it was interesting that it affected mostly like glia type of cells. Like they're becoming more and more important, I think, in the field also. So yeah, I think, yeah, hopefully we'll continue to have a lot of more great researchers like you, and you keeping researchers like you having money to continue this important work. And
Yeah,
yeah,
I have,
come back anytime with updates.
sure. Actually, so I got a position as a postdoc at Stanford, so I'm moving to US hopefully soon, and it would be great to participate and share more about my science. I'm slightly changing my field of interest from my PhD work. Now I'm gonna look at more fundamental questions and study more about how the genome organization works. And one of my most uh, interesting topics I like is heterochromatin, and what's its role in genome organization. So I hope, and that also has a lot of interest, especially with cancer and other diseases. Uh, so I'm very excited about the next phase of the science that I'm going to take.
Wow, congratulations! That's so great. Yeah, I had but it never got funded. Like back in time, I wrote hypothesis paper, and I wanted to look at eye motifs and G4 formations, and look at if it's involved in synaptic plasticity. But I applied for this uh, exploratory
Right.
grants, but it was still too exploratory for them.
Uh,
So. And how it could be involved in addiction, because I was also working a lot in compulsion and addiction related. But anyways, yeah, I wish you the best for that your visa. I had to come with the visa too with my whole family. So I hope everything goes smoothly. I'm sure it will. And yeah, you are always welcome to come back and give us updates, and yeah, and just come also maybe you were interested in listening to some of our guest speakers and how, discuss and comment on work they're doing. So, but Serena, you wanted to say something.
Oh, yeah, just thanks again. I was going to sort of take it off into some general area, but no, it'll be great to see your, and I'm always fascinated about structural organization of the chromosomes. So that's a fascinating area. And I wish you the best of luck, and I hope to see you back.
Thanks a lot.
Okay, so yeah, thank you so much, everyone also in the audience, and everyone that asked questions. And thank you for joining our room today. And please follow the Science Society. We have actually tonight, we have more guest speakers coming a few times, like a bunch of times a week. And tonight we have another interesting topic. He's a researcher at NIH, and he will talk about his latest publication on how mitochondria act as microlenses in cone cells. I think it's a really interesting topic, and we have more rooms like this. And yeah, again, thank you, Monica, so much for coming here and teaching us about your work. It's great, and good luck for everything you'll do, and hear you back soon, hopefully. Thank you.
Yeah, thanks a lot again. It was really great to talk on such a platform. I think it's very important that we learn to communicate our work in different ways, and I can tell you that it was a challenge for me to talk without having a presentation, but also to talk about it in a more general context, and hopefully next time I can do a much better job. Um, but thanks a lot for this opportunity. It was really great, and I'll also be looking forward to participate as an audience for other talks.
Yeah, great. You did a great job. It was great. And yeah, we always keep the recordings here on Clubhouse, but I also put them on the website and on YouTube and on Spotify. So I will send you the links so you can share with people that you would like
Yes.
to have it. And yeah, and it should also increase the altmetric impact. That's why I'm doing to put it on different platforms, because the altmetric I learned is made out of different categories. So having, for example, a YouTube video and a podcast brings up the score than having like 100 tweets always of the same. So I'll also do that and send you the link. And good luck for everything. And thank you.
Thank
And
you so much.
yeah, thank you everyone in the audience, and hear you back later tonight. And bye everyone. I'll close the room in 3, 2, 1. Bye.
Take care.
Bye.
Bye.